Selective autophagy promotes compartmentalized cell elimination under stress. Programmed cell death is important for development and homeostasis and is commonplace and predictable. Morphogenesis, tissue sculpting, and removal of damaged cells is driven by cell death. Conserved cell death in morphologically complex cells, such as neurons and glial cells, is poorly understood due to different parts of the cell that can die by different mechanisms. Conserved autophagy in complex cells is similarly poorly understood. Selective autophagy is the process by which specific targets are isolated for degradation to preserve the cell, under both basal and stressful conditions. P62 is a key player in the process. An isolation membrane expands after binding to sequest1 via LC3-LGG1. It then closes up to form an autophagosome and lastly combines with an acidic lysosome to degrade the target as an autolysosome. With that said, our objective is to test for potential links between selective autophagy and compartmentalized cell elimination using a model organism. Compartmentalized cell elimination, CCE, is a novel and likely universal death program. This novel program, named CCE, was discovered in the nematode C. elegans and is pictured in the two cells below, the tail spike cell and the CEM neurons. A membrane GFP was used to visualize CCE. Molecular regulation of CCE was identified using several CCE screens. CCE utilizes core apoptotic pathway proteins, including caspase SED3 and BCL2 SED9. BCL2 SED9 is regulated via novel proteins in CCE, including BLIMP1 and DRE1. Because autophagy can be induced under stress, we developed a heat shock assay. Over a period of 6 weeks, we used a water bath set to 33 degrees Celsius and shocked NGM plates of young, healthy embryos for 1 hour and followed that by a 5-hour recovery period in the 20 degrees Celsius incubator. We then scored the GFP tagged animals using an upright microscope, looking for any remnants of the tail spike cell that failed to die normally. Various temperatures, embryonic life stages, and plate conditions were tested. We ultimately used the assay mentioned previously. Analysis of the results led to sequest1, the worm version of P62, being identified as a promoter of CCE under stress. Under normal conditions, both wild type and the sequest mutants showed no difference. However, under heat stress, only sequest1 mutants displayed defective CCE because autophagy was not functioning to assist in clearing the cell, which led to our hypothesis that sequest1 may target anti-death mechanisms in CCE. The top diagram here depicts the canonical core apoptotic pathways described previously. The bottom panel depicts BCL2 SED9 as an anti-apoptotic pathway protein. Sequest1 could similarly be targeting BCL2 SED9 and allow for CCE to proceed normally. Mutants of sequest1 may cause increased BCL2 SED9 function and could mean CCE regulation under stress is conserved. BLIMP1 transcribes SED9, while DRE1 degrades BCL2 SED9. They are negative regulators of BCL2 SED9. In stress, BLIMP1 and DRE1 could potentially be downregulated, leading to the upregulation of BCL2 SED9. In our hypothesis, sequest1 is a candidate regulator of BCL2 SED9. We conclude that the data is significant enough to suggest a novel link between selective autophagy and CCE under stress, and sequest1 could be a potential regulator of this stress-death pathway. In the future, we aim to answer how sequest1 assists CCE under stress conditions. The next steps in the project include Gibson cloning, injections, and integrations. Comparisons between C. elegans system studies and other systems, such as mammalian systems, will further help elucidate novelty of our hypothesis.